Okay, good. Today we are going to look at clustering algorithm, and we will first play with the built-in functions in MATLAB Toolbox, statistic toolbox provided, including k-means, dbScan, spectrum clustering. And we will use example data they provided. And then we will apply them to this algorithm to real single-cell RNA-seq data. So before that, let me make that clear. So what we are talking about here. So let's say if you have X matrix, that's what we have here. So what we are going to do is first to do dimension reduction to get S matrix. Basically, that's the coordinates of your 3D where the cell was put in. So by default, we have a gene by cell matrix, but in this S matrix, the cell and the cell has 3 positions. So this will be number of cells by 3 coordinates X, Y, Z. So that's what we know how to do. We can use PCA. We can use t-SNE. Okay. Then we can do the k-means, which the algorithm for clustering. K-means, for example. Okay, so that's our series of this dbScan, spectrum, clustering. So you put this number of cells by 3 matrix as input to give this clustering algorithm. So this will give you the output. It's a vector of your cell, number of cell, and its clustering ID. Okay, depends on how many clusters you want to get. So that's our simplest way to do the information flow. Okay, you use PCA, you use dimensionality reduction to compress your original big data matrix into a 3-dimensional description of each individual cells. So based on that information, you do the clustering. In that sense, you throw away a lot of complex information. You only rely on these coordinates of the cell. Okay, so this is one way. Another way you can do is to use this X directly. So we can transpose this. It's still T, it's X transposed. Now you have a cell and then their gene expression. Okay, so this matrix, transposed X matrix, is much larger. So it contains all the information about each individual cells, their all thousands of genes expression. But you can still trade this as your input data and give the k-means, give the dbScan. Okay, it should work. So once you have this membership, you still want to visualize them, then what we did is put in the 3D using the S information from this matrix. Okay, so the matrix here, the information was used to put your cells in different position. Okay, and then you can color those cells by its ID. So the ID information will be used to color the cell. And if you go the pathway, the analytic flow chart with the pathway on the top, you will get very good separation and this ID because you did the k-means, use S, and then you label the position, use S, put the position. So that will give you the best solution visually. So it looks like, okay, this is a cluster, and then this cluster has been classified as one group. Well, if you go this way, you cannot guarantee the position of S where the cell located is consistent with your membership you assigned, because here the information is different. Okay, you know what I mean right now? So this S is from big X. This X transpose is also from big X. Well, the k-means, if you use S to do the k-means, you get the position and its membership highly consistent, where the location and where the membership should be. Well, if you use the transpose big X here, do k-means directly, you cannot guarantee you have a beautiful separation of those visualization. The position of a cell and their membership should not be consistent oftentimes. Where can we suggest to use the second one, the transport? Yes, so if you do this directly on the result should be highly consistent, because S, based on the dimensionality reduction, based on the cell's distance. Well, when you gave k-means clustering algorithm this transpose X, k-means algorithm also use the distance between cells to classify them. So largely should be consistent. What I described here is there a little bit information offset, which is not consistent for some cell membership. Okay. So in general, the flow chart on the top this way, this means should be fast, because you compress your cells variables to describe cells into just 3 or 4 dimensions, where the bottom one should be slower. But just keep that in mind. We will try those different parts and just see what we get. Okay, use the real data. Okay, come back to the toolbox. I suggest you reinstall it, because there's some update recently, especially for the function. You can select a group of cells and say, I want to create a new cell type for this cluster of cells. And this function become available yesterday. I updated the function to allow you to do this. This is because Dr. Wang requests that function. Say he want to select a group of cells. Say, okay, I want this like a cell type. So it's I show you this function right now. So this. So I use this process data to show this new functionality. What I want to show you is this toolbox is highly dynamic. It's always updated. So for instance, you want to make let's see, let's cell type now. Okay, for example, you want to make this green fibroblast, their subset of fibroblasts as a new group, new data type, cell type. You can select this and see, I can click this button to add a brushed cell into a new group. And you say, okay, I want to have a new cell type out of this. So you can give a name or not. Okay, now you have 6 and 7 become 2 separated cell type. And then if you select to show cell type again, now you see this new cell type has been annotated. Depends on your selection of the group. Then you can rename, say I have a new cell type and I want to rename it. And then you click this button to rename the cell type. New cell type into fibroblast number 2, something like that. Okay. You can always click this button to rename the cell types because annotation sometimes not so accurate, but based on marker genes you observed, you may rename some of them. How can you merge 2 cell types? So if you want to merge duct cells with Mueller cells, you think, okay, it doesn't make sense to separate them. So how can you merge these 2? Do you know? Yeah, so the simplest way is just rename Mueller cell into duct cells, then this should merge. So what you can do is to select the cell type, rename Mueller cell into the same name of the then these 2 groups should merge. Okay. So if you don't have the latest version, you don't have this function, right? I found that there's one cell which in a different color. I want to know what it is. There's one green, orange cell in this cloud. I'm just curious about this. Oh, there's a Mueller cell. Okay, there's a particular cell type which for this particular cell which have the okay, any questions? We are going to do the k-means. Okay. So as I said, we are going to use the built-in function and their examples to look at the usage of the k-means algorithm. So I'm going to just doc means. So this function takes basically 2 input variables, x, big X. Remember, x is transpose x in our case, because that's the samples in the rows and the variables to define each sample's column variables. So make sure you know what's the input supposed to be. And the number k is required. So you have to give a number k as the super parameters for this algorithm. So we can jump to the first example. Okay. What you can do is just copy this and paste into command window, and you should see the data points. So this data set is some different species of fish, and so they extract the data from a table, and the X contains 150 individual fish and their measurement of petal length and the width. So 2 variables to define each sample. Let's see, visually, we can see, I would see there are 2 large clusters, maybe 3 small clusters. So I can see this. Next, see what they did. So they set a random number generator, the seed, to 1. If you do this, you will generate exactly the same output like this. Okay. To make this pseudo random number generator to the same states. So we can, on different computers in different days, you can generate exactly the same random number series of random number. As the k-means start from a random position, so each time you may have slightly different results. For reproducibility, so reproduce exactly the same results, you can follow that. So we can do this. Right, so this function is very easy to use. It returns 2 variables, 2 outputs. One is this ID, index. The index is this ID. So to tell you, your sample, in our case, the sample is the cells, and each ID is the number of this cluster. So ID should be either 1 or 2 or 3. So let's see, the ID is 150 because your X has 150 samples. So this ID contains index. So what that means is the first sample belongs to cluster number 3. If you move down, you should be able to see, okay, fish sample number 65 belong to cluster number 1. And there are 84 cluster number 2. Okay. So what's the capital C here is the position of the centronoids, I believe. Yeah. This is not very important because that's it found the centronoid of these 3 clusters defined by X and Y because we have 2 variables. Now, how can we visualize this? Okay, now we are going to color those data points by their membership ID. We can use different colors to code those and then to look at their membership. Okay. What I'm going to do is to use my own way. It's figure. I'm going to use scatter. Oh, no, let's do, yeah, scatter should be fine. So I have X position 1, X position 2. And then the next is the size of this marker, and then IDX. So this is a way I'm going to use to plot samples. Okay, so this is also the way I plot those cells in my applications. Just use scatter plot and gave the index. The index will look at the 1, 2, 3 as label. They're not real numbers, they're just labels. So it doesn't make any comparisons in number. Group 3 is bigger than group 1, it's not, it's just labels. And they label the cells, label the samples by their color, by ID, okay? The function called scatter 3 that will have 3 XYZ positions, then otherwise the same. Any questions for this? And also you know how k-means algorithm works internally. It's just randomly put a centroid first, then try to find membership and then shift until stop. So this is a small example. Use 150 samples, 2 variables, and to do this. Let's look at this documentation. Yes. So this is a little bit complicated, but if you like, you can try those code. So you have a group scatter, the function, and then you can generate something like that. But in general, this should work, okay? Another function I want you to have a quick look is k metanoids. I don't know this word. So maybe you want to read the documentation by yourself and spend 3 or 4 minutes to see what it is and what's the difference between this function with k-means. You can even use this function to replace k-means to do the same thing and see whether you get a similar result or not. So try this yourself. Metanoids. Okay. So I have 2 figures on my screen. Figure 1 is on the top, is a result generated from k-means. And figure 2 is using this k-meanoids K-mean noise. So that's a documentation for Medois, K-Medois clustering, and there's one section I believe explain the difference explicitly. So that's a difference. So it's similar to K-means, and as we tried out using this small example, the outcome is almost identical. The only difference is this algorithm return the central noise from the pick one of the real data points from your input. Okay, instead of compute some mean in the middle of nowhere of your cloud, it's just pick one of them as your centroid. Okay, this is very small difference. You depends on your requirement. Yes, either you want this C to be one of them input data points or not. Yeah. I don't see any. There's no much difference. I believe. Yeah. I think a K-means just fine, unless there are special requirement. You want to have the real data point as a central node, which is present in your input data. Yeah. 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 Well, the k means you have to define k. So I'm not so sure you can define a k as 1. If you do that, there's no cluster. You can still run it. So yeah. Now let's see first which dbScan. So this will tell you where this function, the file define this function is located. So it's located in MATLAB program version number toolbox statistics. Inside their statistic method, and dbScan is one of these. Similarly, and you can see a k-means is somewhere where it is located. Okay. Well, if you see which SC3, there's no SC3, but if you see run SC3, it's located in this toolbox you installed. And the top two, this is if you have this toolbox installed. Yeah, this is so-called preload, or it's package provided by MATLAB, the company. And SS3 is a function I wrote myself. So you have to download this package installed, then you can have SS3 application in there. Okay, so that's the idea. So if you know those functions by documentation or other channels, then dbScan, you can see, okay, I want to look at the dbScan function. How can I call this function? It's documentation of it. So this is different from the k-means. K-means have a x and a k, just two input variables. This one, you need 3 variables. You need x, your data matrix, contain the sample you want to cluster, do the clustering. And then you need epsilon and the minimum number of points. All right, so that's what we mentioned. You have to have the circle, the radius of this circle, how big your diameter should be, and in order to define the neighborhood members of the sample. And you require the minimum number in that circle, how many of them, and then you can consider in the middle of these data points, the core data point. So that's remember the tutorial video and explain those concepts. So we cannot ignore this. This is like hyperparameters you have to give to the algorithm. Otherwise, there's no way you can run this program. That's a limitation again, right? You have to define your own epsilon and the minimum number of points in order to get this work. So we can click algorithm, the explanation about this. So all in the same documentation. So what I clicked is this section, algorithm, and then by just reading this technical documentation, you actually can learn a lot because they are well written. Okay, yeah, well written documentation by the expert. So I want to give you 1 minute to read this to clear your mind. Okay, let's use an example and to see how this works. Okay, so they again use this random number generator to generate a specific data type, data set. When you plot them out, it looks like this. So I will shut down my previous window. Again, I'm using the same scatter plot. In this case, we don't have ID. Okay, I only gave, so if you want to make your point smaller, you can say, okay, I want to make 5. This is a small data point. I think you can try to run k-means on it. It's supposed not to give you very good results. So if let's just write k-means, because it's special arrangement of those data points on the plan. Index. Let's see, we have 2 clusters. Okay, let's run k-means, then try to figure IDX, right? So that's a k-means solution by k equal to 2. Now let's try dbScan. I have no idea how to set up this variable, input variables. So maybe I put 1 and I need 5. Okay. So those functions should have a similar structure of input output. Output, I assume, should be this. I just guess. I don't know because they belong to the same group. So the output should be this ID of your member ID into different classes. I just gave it a try. Okay. Then look at the output. Oh, terrible. So I don't know how to use this function. So I have to, how can I control the number of clusters? So it gave me, look at least 3 or 4 different clusters. I don't know in the documentation how it's controlled. Maybe I need to increase 0.5 to 1.5. Let's see what happened. It's better, okay? So this is what we human intuition supposed to classify them into 2 groups. So this parameter will determine the output, the outcome of your clustering. So if you cannot see the data points, you don't even know whether you have a good setup of your parameter values or not. So this is a problem for dbScan. So in the package, I just cannot use the dbScan, although dbScan sometimes it's density based. If you have a cloud of your cell, which highly dense, where another is not so much. But I have no guarantee I can set up this hyperparameter correctly to solve the problem. So I cannot use this function at all. What's parameter they used in the example? Yeah, so they use 1 and 5 to do this. They use this group scatter to give this plot. So you can try this. There are other parameters we can use. For example, you can select the algorithm to compute the distance. Okay, this, you know, the lecture we talk about distance. There are Mahalanobis distance, there are Euclidean distance. So sometimes different distance measurement give you better or worse results. But in general, we use Euclidean distance metric. Yeah, so they also show, okay, if you use k-means to get the k-index, not as good as we want. Here they have a more complex structure of the data. Okay, any questions so far? We're fine. Okay, I'm going to clean this, close. Oh, let's try spectrum. So the last algorithm we mentioned, which spectrum class 3. So again, this function is inside statistics toolbox. Let's see what the example data they created to show this function. 2 inputs. X and K, okay. So if you gave it the X matrix, which your original, your raw data matrix, they will compute pairwise distance between your samples. If you pairwise distance already computed, you can provide this distance matrix as a pre-computed S matrix. And internally, once this distance matrix between samples has been computed, the algorithm will create a network out of it. Either this distance matrix is computed or provided. So based on that network, it will generate adjacency matrix and then compute the degree matrix. And then adjacent degree matrix minus adjacent matrix becomes Laplacian matrix. Then eigen decomposition on that. Then k-means on the eigenvectors, which is smallest. Okay, so example data they use is the same as the one we had. So in that case, we can just compute. So I already have the old one in my memory of here. So I just directly use this in the figure. Okay. I just demonstrate it. It's okay. Same as the DB scan. But you don't have to give the the length of the radius, and also don't have to give the number of points, not like a dbSCAN. The two extra parameters you need. Okay, so this will work. This works, okay. Any questions for those general walkthrough of those major clustering algorithm? So when you come back after this 5 minutes break, we are going to use real data, single cell data, to plug in this to replace this X. Right now, this X is small examples. But let's use real data and see what happens. Okay. Okay, so far this big X small example data just directly provided by those built-in function documentation. Replace those big X data set using single cell data, okay? So we will use S as our big X to give to algorithm. We also use our real UMI count 2 different ways, okay? Let's clean this. Let's see clear O. Now, what we need to have is this example data set. We can use processed data because we want to have the t-SNE coordinates. With this figure, let's click this export and save data and select workspace. We will need big X and S small s. Okay, so we will use small s as our input for clustering algorithm. We can also use big X as our input. Any questions? What we are trying to do? Yeah. So if you have a clean workspace, but after you click this, you should have 2 variables. So in this case, I can shut down all the other windows because I'm going to working on this 2 variables, two input for my cluster. Okay. So this is a matrix, small s matrix is 2075 cells and their position. Okay, to visualize them, let's do scatter 3. Now it's not X, I will use S1, S2, and S3. The rest of them just, maybe I put a 5 here to indicate the size of my marker in the plot. Okay, now you can see each cell is defined by 3 numbers only. So we don't have an expression, we don't have a name, genes, nothing, just position. Okay. But by looking at this, I can start to count how many K I want to have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, maybe 11. Okay. Now I'm going to apply this k-means. Now my input is not a big X, it's s small s, so I have 11K. I hope this makes sense to you, and because we are doing embedding based. So you get the coordinates of the cells and then just rely on their position to compute the distance between cells, nothing else. So this k-means give you the membership ID. Now we can replot this scatter plot 3D, but give the index. Okay, before there's no index information, all the cells have the same color. But now you have 11 different colors. So just that easy, okay? So I will work around and see whether we get a similar plot. And then your job, if you finish this, your job will be using big X. See, can you do the k-means for the big X? And then replot the color for the membership for the cell. So we'll work around it, make sure. Makes sense, right? Okay, good. Yeah. Yeah. Okay. Mm-hmm. Let's see, do you have S? If you don't have S in there. Oh, yes. So this S is not what we need. This S. Let's see. You have to redo this. Then export workspace. Uncheck this, select this and S. Okay, so now I save them.
Oh, we converted S into X?
Yeah. So the S here is the position of all the cells by their XYZ axis value. So now you can do, yeah, you can work on S. No, we don't have that. Oh, good. Yeah, that's good. So this color bar spectrum is designed for a continuous variable. So you have 1, 1.5, 2, 2.75. It's not a categorical, but you can make it like steps using different color map. But yeah, in this case, it's fine. Yeah, that's beautiful. Oh, you tried the big X. Yeah. No, the big X is not big X is not the coordinates for. Yeah, this is big X is expression level for a particular gene. So yeah. No, you just cannot plot the big X. Yeah, good. Yeah. Sure, you don't need help, right? So those clusters are separate clearly between the groups you see on this 3D plot. This is because your clustering algorithm is running on those coordinates of the cells in the small s variable. Let's see, I just keep this still here. So let's see what we can do with big X directly. So I'm going to do some normalization transformation. Let's see, normalized. Let's see, norm. The reason I want to do this is because internally the k-means compute the distance between cells. The distance we use is Euclidean distance. Euclidean distance works better if you variables, those gene expression level follow Gaussian distribution. But right now, because we X is row UMI counts, it's highly biased to a lot of zeros first, and they definitely not normal distributed. But if you do the norm transformation and also do logged, so let's see, log xn. So I will basically take a log for normalized +1. So do some transformation will help to improve the accuracy of the computation for the distance between cells. Okay, with this transformation, I'm ready to give my input to k-means. I still ask for 11 clusters. I have to do this transpose. The reason, don't forget this. Otherwise, you will do a k-means on the number of the genes. Okay, so you transpose, make sure your cells is in a row. Well, I have no idea how well this will be. So I will plot the same, use the same plotting function to look at. Compare with my figure 2, which is based on the coordinates itself. And figure 3 is based on distance between cells. Okay, do you see the difference? Yeah. On your left, you gave the coordinates of those cells to k-means. So k-means will look at the cells and compute their distance purely based on their 3D coordinates here. Well, the distance you compute from big X is considering all the genes expression profile and compute the distance between cells. Yeah, so I will work around and look at see whether you have any questions. Yeah. Which one? Yeah. The log transformation doesn't work. Do you have the variables here? No, this log, you have only 11. I don't know how you did. Yes. So your big X becomes only 11 and 2. So yeah, you probably want to use the interface to export your variable again. Mm-hmm. Yeah. Now you have SX. I don't know which one you have. You probably have X4 is your current. Yeah. So maybe you want to do that again to overwrite the you click this and the workspace and select big X. Big X is yeah, and replace X5 to X. So it's override X. Yeah. 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 So now this big X is correct version. Then you do this normalization and then take a log. Mhm. Let you know, you use different input and get different cluster outcomes. Yeah.
So whenever you enter a function, like whatever we previously, how to do that? If I need to like do this function,
Yeah.
what is the method to do?
To do which one? Which one?
If I always used to just copy and paste it. Is there a way to do that? Like in a shortcut or something?
No, that's just the same as what I used in the
Like if I want to like type the exact way, is there a shortcut to like give up this specific command?
oh, how to get this? Let me see. So if you do the k-means, then if you don't know which one next, you may press the tab key. I don't know if others, yeah, the tab key.
Just
Tab, yeah, after the comma, the tab, and then the other variables will, yeah, let you to select, yeah. Mm-hmm.
I should have just.
Yeah, it try to help you to fill in some of the possible options. The tab key is a good help. Yeah. So so you know how to do k-means with this format. You can try spectrum clustering. You can try which other dbScan maybe. I don't know. Yeah. Spectrum clustering will be able to handle such a big matrix. Remember, this is supposed to be okay. Yeah, give it a try. And so remember, they take the same format of the input. I hope you'll be done in 3 or 4 minutes. Yeah, so we run into some problem because, let me think about this. So we have Laplacian matrix, which not symmetric there. There's some some problem with this eigen decomposition and gave you the complex number. Okay, so the k-means internally won't work. But if you do this on the s small, this should be okay very easily. So that's a spectrum clustering based on the coordinates. So the last algorithm I'm going to show you will be this SC3 run SC3 let me check. So what I want to do is to look at my original code. See, I do have a k as a second variable. So okay, so this function will take care of those normalization log transformation by itself. So you don't have to worry, just give directly the x. Okay. Remember, you see this flow chart of SC3, you have 3 distance matrix and transformation into each one have 2 transformations, then different. So that's the different steps in showing in there. By the way, I do put this reference, this paper in the nature method described as SC3, this reference. Yeah. So many of these published methods, if you read their methodology from the paper, you can code by yourself, but just follow the ideas. Many of them is very simple, actually. If you know, okay, each steps, this is like a puzzle, do this first. So you can re-implement the whole package using, in this case, SC3 a little bit complicated. So I used some, this particular function, but for many others, just a couple of lines of MATLAB code. Yeah. Mm-hmm. Let's check you. Oh, is this really? Get rid of this. Save. Well, I don't know what's wrong with this.
Exactly.
Uh-huh.
Because the SC3.
So did you change this function?
No,
No, I
I
didn't.
just opened.
Why it's
I just
running?
click to see what's wrong with it.
It's running okay on my computer. Let's see what's the error message. Well, I really don't know, and you maybe want to re-download that package. Yeah, but it's okay running on. I never had that problem. You don't have that problem. Yeah.
No,
I don't know why some source code have been.
it's not
Change.
being recognized here. Yes,
Mhm,
this doesn't
mhm,
count.
mhm.
It's not
Yeah, you can just reinstall the package and override that. Yeah. Yeah. Yeah, on my computer, this is finished. So I have this index which computed from SS3, so I can run this figure again. So that's my solution from SS3. 11 members of cluster. Okay, so side by side, we can put quite a few of solutions. So that's number 2, number 3 is a k-means running on the big X. And number 4 is my spectrum clustering running on coordinates. Number 5 are the SA3 clustering. Okay. As I said, statisticians like to do clustering methodology development. But for single cell, it's really, there are different ways to cluster cells. At the end of the day, I don't think that's very important. You can cluster them as the way you want. Okay, so if you read another paper next week, say there's a Nature Communications published a paper, say I found a way to cluster cells into a single cell data set analysis. Don't take it too serious. It's just another way a statistician try to cluster cells. So really, how are you going to look at those cell and the types and look at individual genes expression profiles? That's more important. So you have to have this way to easily to access your data and look at individual genes one by one based on your personal experience. There's no way you can use any clustering algorithm to help you. They're not very useful at all in this case, right? Yeah. Yeah, I can. Yeah, so let's finish this in 5 minutes, then we come back to the practical and solutions for those. Okay. So any other questions for this clustering algorithm? How to use this function? Yeah, I think the rest of this half hour, I will use the real data set, the example data set, to show you, okay, we can do SA3 clustering first, then annotate the cell, and then we can do k-means first, annotate the cell. Let's see what we can get, okay? Some practical solutions. I'm going to close everything. So let's start from this raw data, okay? Just assume you have nothing and you just get your own data set in there. We know the first thing you can do is to do this embedding, okay? Okay, with this, I can run k-means algorithm. So 11. Okay. So this is one approach. So I just keep this window here. So now we finish this embedding, clustering, making separated the clusters. What you can do next will be you can check, say, okay, what the gene express highly in this each clusters, like marker genes, and then decide the cell type. But I show you another way to do the clustering. So again, I keep this window here. I just do another raw data independently, okay, to separate the windows. In this case, I'm not going to do embedding. I just directly use this clustering. I will directly clustering use SA3 apply to raw data. So you only have the big X matrix. You don't have S. You don't have a position. All this position of the cells are the random points. So you can run this directly cluster, use big X, say SA3, 11. So completely different way, right? So I'm going to cluster cells first, then do the embedding. or use k-means. Now I have 11 clusters in here, okay? It doesn't use any information about the embedded coordinates at all. It just directly read out the profile of this gene expression of individual cells, classify them into here. So now I want to run embedding on this. So on this already clustered cell. And embedding algorithm will not use member ID of their clustering members. It just do whatever is independent embedding, right? But largely, you see the solution will be very similar to SC3. So all the orange cells were embedded into the same corner of this 3D space. And the light blue ones are also clustered together. So it's largely consistent, right? I hope you know the difference right now. On your left, the clustering is based on the position of the cells by their embedded coordinates. Well, on the right, I didn't give any information, I only gave their expression profile and let SC3 to cluster cells into groups. Then retrospectively, I embedded them and the solution of the cell distance the same as the cluster SC3 solution. So that's why you see this consistent same group of cells, and they has been clustered in the same group, and also embedding and tell you they are separate class. Okay, with either of this solution, and you can use, see, I want to identify the marker genes. Let's see here. Yeah, so what I can try is to brush this brown cells. I want to find the marker genes for it. Just click this one, this button. This will give you the top 10 marker genes for the cells you selected. Okay, so those are the examples of marker genes we found for this cluster, this cell selected. Okay, I believe if you know the PCAM1 is, I don't know who knows. Okay, on your right side is this like a stem plot to show the expression level for the gene, and then your left side is a heat map to show this expression level by this color bar. So that's the way if you know the biology of those marker genes, that you know, okay, the cell type is what the cell type is for this cluster. Any questions? the week after, next week, we will look at the regression analysis and show you how this marker genes has been identified. So it's using a lasso, so-called regression method. You can also use this function, marker gene heat map. We have 11 subclusters, so this could be a problem. That's cluster ID. Let's see the analysis. Different algorithms generate slightly different solutions, but this is one, and you have 11 clusters. So for example, you have number cluster 7 contains more cells, so you have a wider space here. Assume those genes are the marker genes, but some of these marker genes not so specific. For example, for those, it's also expressed in class 1 and 6, right? The sum is okay. So, but after that, you can look at the gene name and pick manually. You want to put in the report, or you want to show people. This is just one method. You see, you have 3 different methods to select to generate a slightly different heat map of those marker genes. There are other buttons if you want to try, you can click. I don't even remember what they are. So this plot is generated from the same plot here, but it just take average. So you get the same bins, just one single value for the whole average of all the cells in there. Okay, it's a summarized. So this inlet, this picture is the same as this one. Instead, each group for particular gene is average of that gene expression across all the cells in that group. So here, for example, you have group 7, you have a lot of cells, but you take average, you only have one value. So this is like a summary of the larger picture. So the picture underneath looks messy sometimes, but if you use summaries, the picture may be better for publication purpose. Oh, you click this button here. Yeah. there's no cell type annotation yet. It just gave you the potential marker genes, which specifically express in different clusters. For instance, this box here, APOE and those other genes express highly in cluster 1. So with that information, you probably can tell, okay, cluster 1 belongs to which cell type. You can save your marker gene list to the variable in here. So that's your marker gene named. Okay. Yeah, sometimes you use a bubble plot, we will get a better results, but not in this case. Okay, so this is all I want to share with you today. I hope you understand better now how these two buttons works. So you cluster cells by embedding their position, their coordinates, or you can do directly from an expression matrix. Then you have multiple methods you can select from. And this SC3 supposed to be the best one. But at the end, there's no much difference. So the general procedure I told you before is I do this embedding based clustering, and also I over cluster recompute. Instead of to do 11, I probably do 25 and annotate the cell individually, small groups individually, then merge cell type name. So that's basically that's the way I want to do it. Yeah, at the end is you have to check those marker genes one by one to make your own decision. See whether you separate these two into two subclusters or not on your own decision at the end, okay? So there's no machine learning, no artificial intelligence. It's all human intelligence getting involved here. All right. So next Tuesday, we will be talking about the classification. So once you have those marker gene database, how are you going to classify cells to different categories? That's the problem of the classification. Okay, let me know if you have any other questions for this. Okay. Oh, by the way, last week I told you we can run R package from here. So if you want to give it a try in the external here, there are run R Surit workflow. So you can give it a try and see whether your computer setup is working. And you can just click this, and then you can look at the scripts that's written in R and just run it. You can run this on the raw data. Okay, thank you. See you next Tuesday.